Good day. I want to present my poster titled Transcriptional and post-transcriptional mechanisms that regulate gene expression in lung cancer cachexia. As an introduction, cancer cachexia is a multi-organ syndrome where the tumor secretes cachexia-inducing factors that signal to skeletal muscle and adipose tissue, inducing atrophy in wasting. These changes in these tissues are related to changes also in gene expression, but we don't fully understand the transcriptional and post-transcriptional regulation of this process. So our main goal is to assess the transcriptional regulators like transcription factors and post-transcriptional regulators like long coding RNAs and RNA binding proteins that regulate gene expression in skeletal muscle, adipose tissue, and tumor of cachetic mice. We have this study of cachetic mice, both female and male, and the RNA were extracted and sequenced of gastrocnemius, white fat, and lung tumor. And we perform differential expression analysis, gene ontology, motif finding, and RBP finding. Our first result is that these tissues have transcriptional changes in cachetic mice. Here in A, we observe the deck number of the three tissues and showing that adipose tissue have many more decks than the skeletal muscle and the tumor. In B, we observe the gene expression profile of skeletal muscle, showing a very conservative response. In C, we observe the gene profile for adipose tissue, showing a very variable response. And in D, we observe the gene expression profile of the tumor, showing also a very variable response. Second, we analyze the pathways and transcriptional regulation of the three tissues of cachetic mice. Here we observe the results for skeletal muscle where pathways like proliferation and differentiation and ion transport and excitation contraction coupling are regulated. In the second frame, we observe that 22 of the decks are transcription factors like FOXO, BCL6, inupregulated promoters, and IRF5 and TBX15 that are regulated in downregulated promoters. For adipose tissue, we found pathways like thermogenesis and lipolysis, and we find 80 decks that are transcription factors like TCF8, ATB5 in upregulated promoters, and CREB5 and KLF6 in downregulated promoters. For the tumor, we found pathways like proliferation and metabolism that are regulated in cancer cachexia, and we also find 80 of decks that are transcription factors like ISL1 and AP1 in upregulated promoters and TYRP63 and GATA1 in downregulated promoters. Then we perform a correlation analysis between rRNAs and London coding RNAs, and we found for the three tissues that the vast majority of the non-coding genes are London coding RNAs. So for skeletal muscle, we observe 7 significant correlations, and these pair CCN1 and GM that are related to differentiation and extracellular matrix changes. For adipose tissue, we found 183 correlations, and this pair UCN2 and GM that are related to lipolysis. And for the tumor, we found 113 correlations, and this pair of this integrin A and ERIC that are related to stemness, tumor stemness. At last, we perform an analysis or RBP regulation in cancer cachexia. We found RBPs only for adipose tissue and tumor, and these RBPs are related to binding and protecting nascent RNA, splicing, and alternative splicing. The expression profile show a very variable response of these RBPs in adipose tissue. And for the tumor, we found 10 of the decks that are RBPs related to alternative splicing, regulation on RNA stability, and alternative splicing. And we also have a very variable expression profile. So our conclusions are that the activation of transcription factors contributing to muscle atrophy such as FOXO1, adipose wasting as TCF8, and tumor progression as IAP1. Second, we found post-transcriptional regulation by London coding RNAs of genes related to myoblast proliferation in skeletal muscle, lipolysis in adipose tissue, and stemness in lung tumor. And third, we found post-transcriptional regulation by RBPs, such as MKRN1 in adipose tissue related to browning, and NOVA1 in tumor related to tumor progression. Thank you so much for your attention, and I'm available for your questions.